Yes, so there is in the slides, yeah, that one. So there is this slido.com, and then there is this number 3669532. And when you go to the Slido, then just put this number there, and the window that we saw previously, that will open, and then you can type a question there.
Thank you. So take the number and go to Slido or ask live. So we will next have 3 short talks, 9 plus 3 minutes. Stick to the schedule, please. So the first speaker will be Professor Laurent Gatto from UC Leuven, and he's professor in bioinformatics and very long time member and contributor to the Bioconductor community. Thank you, Laurent.
Yes, Johannes, you're right. My Emacs slides exported to GDF.
I don't know how to open it big.
But how do I go from download it? Or because if I click here, it kind of scrolls down, which makes it. Well, in the meantime, so my name is Laurent, and I live in Belgium. And Belgium is a really interesting country for the best and the worst. I mean, the worst is sometimes we don't have a government, and then sometimes we have a government, and it's even worse. But Belgium is also sometimes a country that is at the forefront. So, for example, in terms of open access, we have a law that forces us to publish our work open access, and the publishers have nothing to say. And then there is a second interesting fact about Belgium. About 24 years ago, on the 28th of May 2002, Belgium created a legal framework for assisting dying, for euthanasia, second country in the world after the Netherlands, which might be related or not to my talk, you'll judge, which is the tragic death of open source research software. Could you move the screen thing on the side, because my content is at the top, or move it at the bottom? The bottom is all right. Okay, so let's start by set the stage. Fantastic. I think we all agree here in this room that scientific software, is a central player in modern research, biomedical research and so on. That's maybe good, but that also means that it's a single point of failure. Okay, so here's our use case. So we have set the stage. Here's our use case. Imagine you are a third-year PhD student in biomedical sciences, and you study a specific disease. You want to understand the molecular mechanism, behind this disease, and you have set up the perfect experimental design to answer your question. It took months and tens of thousands of euros to generate your multi-omics data. You even came across a paper, that describes a very similar approach, and they even provide software to analyze your data. I mean, at this point, your results are at arm's length, right? What could go wrong? You have your data, you know it's of good quality, and it's clear in your mind how you have to analyze it. What could go wrong at this point? Well, unfortunately, lots of things could go wrong, and we often call about software rot, software collapse, or software death. And this means that software will eventually fail or die if it's not actively maintained. Even if you don't change your software, other dependencies can evolve, and your software will fail eventually. And this can be, due to bugs, or even if you change your software, accidental or voluntary changes that will make your software evolve and change, and for example, not being backwards compatible. So what could go wrong? Imagine you want to find a software to analyze your data. Maybe the software is not there anymore, especially if it was available on request. Maybe you find the software, but maybe you can't install it. And we know that with complex software, installation can be difficult. Maybe you can install it, but maybe it doesn't work, or there is no documentation, no test data. I don't really know what to do with it. And even if it works in the absence of additional artifacts, how can you really judge whether it gives you meaningful results? So, lots of things can go wrong. The results are not quite there yet. And now you might wonder, but really, Laurent, are you hallucinating, making all these things up? I'm sure some colleagues in the audience will agree with me that, using software is not straightforward. And I'm not aware of any research that tries to systematically assess software death in research, but I recently came across the Weekend at Bernie's a blog post where they showed that a substantial amount, or a significant amount, of open source software, so not related to science, but general open source software, were different states. And so they defined these different states as being active, and so still maintain, or inactive, or dead, or even of unknown status. So what do they describe here, this software dying? Some of the examples that I think we are very familiar with, the maintainer has disappeared, has moved on. What happens when a master student, PhD students moves on, or even they are still available, we often get buried into a lot of work where maintaining the software becomes a side project. They also talk about softwares that get split, forking, and that somehow I think happens also in our cases where initial software is released under an open source license, and then suddenly the researchers think, oh, actually this is quite widely used. Maybe we should close source it and make a business out of it. Or broken pipelines, broken dissemination pipelines. And I think this also happens for us. Imagine you have your R package that you disseminate through Bioconductor or CRAN, but these software also live on GitHub. So I would be curious to know how many softwares get updated on GitHub on personal repositories, but never make it to the official channels. And so in that study, they counted that they have between 5 and 20% of software that are dead on the different channels, whether it's Cargo for Rust, PyPy for Python, and so on. And I think that, I don't think we do any better in research software. So we certainly do, along these same lines. So what can we do? And I think we can reflect along 3 different lines. The first one is, do we really need new software? The second one is, what can we do to improve our software survival? And then the third one would be to think about retiring some of our software. I have no idea what I'm doing in terms of time. So tell me when I have 1 or 2 minutes left. So do we really need new software? I think if we think about software as an investment, so of course there is short-term investment, that's the maybe fun bit where we write code to develop. But I think there is also a long-term investment, with the maintenance and the maintenance burden. This can be a considerable amount of work. And also when we think about new features, or features that we want to add to our software, there is this concept of feature creep, where we want to add things to the software, but they are not really, they shouldn't be part of it. So before writing new software, I think it might be worth considering the, science we write reviews. Reviewing a set of software for a specific field, not necessarily benchmarking, we're trying to find out what is out there. This is also what we ask nowadays for new packages in the vignettes, new bioconductor packages. And, you know, would it be conceivable to contribute what I need to an existing software, an existing collaboration? And here is, of course, where open source and collaborative software development become fundamental. And then, you know, that also allows us to join the community rather being kind of a lonely developer. Okay, so we need to write our software, but what is it about the kind of long-term maintenance of our software? And of course, here there are lots of kind of technical aspects where we can make our software more sustainable. And we could talk for a long, long time about all these technical aspects, you know, writing modular software, testing our software, continuous integration, writing good documentation, version control, and so on and so forth. And I think we are here at the Bioconductor conference. Bioconductor does offer, you know, infrastructure and facilities to help us towards writing better and long-term software. And I think planning about the retirement of our software is, I think, is also really important. Think about the software life cycle. So as I said, there is maintenance, you know, feature creep, but there is also possible to remove features. And I'll show in a minute how to do this in R. But also thinking about pausing our software or even handing it over to somebody else. So when it comes to removing functionality, so there is the chapter 28 in the development maintenance and peer review book on Bioconductor that illustrates these two ways to defunct. Is that the timing signal? Okay. So you can kind of defunct or deprecate functions to tell users that soon the functions will be removed, or that the function is not available anymore, rather than completely removing them. And these are two examples from a rather old package. Shutting down projects. So in Bioconductor, there is a kind of a handing over page where you can say, please help me maintain your package, or is there somebody that wants to take it over? There is also an end-of-life policy for packages, just like we have for functions, deprecating and defuncting. And then, of course, I think it's, even if we want to remove our software, it's really important to archive it on Zenodo or the software heritage, which are really, I think, really important projects to, even if we stop developing and maintaining our software, to somehow keep it out there, you know, for archaeological work. Of course, we want to make our software live longer. Training is extremely important. You know, conferences, workshops, hackathons, here are a couple of examples. And also we need the right incentives. You know, what are the career paths to maintain software? There is money for software, but generally it's to develop new features. There is very little to maintain software over time. And we know that we rely on lots of software. But I think these options are very often difficult to implement, especially for junior researchers. So that's something where the more senior people in the audience and in the labs should kind of reflect long-term. What does it mean to have these long-term investments? And as I said, the incentives are often rather novelty, whether, you know, it's papers, software, we want new things, and we don't consider enough the long-term impact of this. And I think the problem of software collapse and software sustainability, and there is the software sustainability in the UK that can help with this, is just as much as technical, how to write better software, but also community issues. And the incentives are not with us, but I think reflecting of what we can do is probably a right step in the right direction. So everything I have said much more eloquently with further details is available online. These are some of the papers and blog posts that I read to help me think about this. And I think there are lots of opportunities to discuss about these topics. So if anyone is interested, please come to me or other senior colleagues. And we could even consider bird of a feather, you know, discussion on Friday on any of these topics that you might be interested in. Thank you very much.
Thank you so much. So, question?
Hi, thanks. Daniel Laila from University of Helsinki. Good pointers. One thing you didn't touch on, and I think it's coming also to Bioconductor, what's your stance on AI slopware? Because I think it's coming, and it's hard to distinguish from good, well-designed software engineering versus somebody who doesn't know how to code, but now has access to cloud code or something.
Yes. Where should I begin? So, first of all, Bioconductor has an official policy in terms of AI contributions. It's chapter 19 in that book. It's very recent. And the policy, in 2 words, says that you should be responsible for the code that you submit. If you submit your packages, you should know what it means. And you should also make sure that you can redistribute it license-wise. And this is true for code that you copy and paste from the internet, and that is even more so using, Gen AI or agents. So that's kind of an official policy. Yes, I think making it much easier to produce code will likely increase the risk of having more software that is arguably more difficult to maintain. Now, having said that, we don't need to publish all the code that we write. So if people want to use any tools that they see fit for their own work, that's fine. But I think we should then refrain ourselves of wanting to kind of submit it to CRAN Bioconductor, which implies work by other people that review the packages, building, checking these packages every 24 hours. So these are additional costs. So if people want to use them, okay, but it's probably an even better reason to think about what do you want to achieve with this code, and maybe to keep it for yourself or your own work and not necessarily release it and then kind of increase some of these issues that I just discussed. But yeah, that's a really good point. Thank you.
Thank you for the talk, Artur Sannikov from University of Turku. Very minimalistic slides, I really like those. So it's not just data, it's not just software road, it's also data road. So many supplementary materials is being shared with Excel files with some particular formulas that are being used there, and a lot of supplementary data is shared with docx files, which if you open in LibreOffice, good luck if it's not all over the place. So how do you solve these issues, not just software?
Yeah. I think there are lots of efforts in terms of data management plans, repositories specific for types of data or more general like Zenodo and others. Yes, you are right that lots of data is being produced and maybe not reused enough. I think efforts in data management and training in data management can help with that. Yeah, it's a good point, similar for data.
Thank you. While the next speaker is preparing, I would take the last questions from the slide. There is what level you think long-term maintenance should be addressed, university or government, for instance, and then also the licensing question. Do you have view on the licensing, which license to use?
Okay, what level should we do? Yeah, that's a good point. I think community level probably first. I think universities or funding bodies should also invest in career path and research software engineers, people that can maybe step away from the novelty and the publications and invest time in maintaining software. But I think in terms of communities, maybe we don't have the power to fund long-term positions like this, but we can train young researchers and developers. Licensing, I think that's a long story. We can discuss this maybe around a drink.
Thank you very much. I agree. So yeah, thank you. We went slightly over time, but we also started a bit late, so that was intentional. Our next speaker is Liz Ing-Simmons, and I did a PhD at the Imperial College in London. Biologist turned into computational scientist, and will speak about sustainability in computing, if I'm correct. So welcome. And I will stand up 2 minutes before you have to stop talking. Thank you.
Okay, thank you for the introduction. Yeah, my slides are HTML, so hopefully they're going to work fine. So yeah, great. Let me check that this also works. Perfect. Okay, so different type of sustainability now, environmental sustainability. just to add to the introduction, in addition to being a research software engineer at King's College London, I'm also a NetDrive champion. NetDrive is a UK research and innovation funded project, which is all about identifying and promoting pathways to increasing environmental sustainability in research computing. So how do research computing and computing in general impact the environment? There's a number of different ways. The most obvious is probably through energy use. Data centers use large amounts of energy, and this is predicted to increase massively over the next few years, mainly due to demands for things like AI. Data centers also need large amounts of water for cooling, and this can exacerbate water stress and drought. And data centers also need hardware to fill them. So mining minerals to produce components for this hardware can also impact the environment, and disposing of that hardware at the end of its lifetime, if that's not done properly, can also have adverse effects. So it might seem that our individual contributions and our contributions as bioconductor developers are quite small, but I believe that we can all have a part to play in reducing these environmental impacts. In particular, I think as researchers and open source developers, we have a lot more flexibility to innovate and come up with novel solutions, new ideas, and ways of doing things compared to people working in industry who are under kind of different constraints, different pressures. And for Bioconductor in particular, Bioconductor is a really welcoming environment for novices, novice programmers. I know it was for me when I started out. And I think that means that we have the opportunity to set people up with good habits, more sustainable practices at the very beginning of their programming careers. So what can we do to reduce these environmental impacts? The Green Software Foundation defines 3 green software principles. So the first is energy efficiency, so using less energy to do the same task. The second is hardware efficiency, using the hardware we have as effectively as possible and avoiding buying new hardware. And then there's carbon awareness, so making use of renewable energy where we can. I would add an extra one to this, which is sufficiency, doing the minimal amount of computing that's required. I'm going to talk about these in terms of which ones apply to the developer workflow and then which ones apply more to user-facing code. So starting with the developer workflow, the principles that are most relevant here are sufficiency and carbon awareness. So sufficiency might look like using minimal examples in your tests and your vignettes, the smallest possible example that's still useful, which is probably something that most people are already thinking about. But it also might look like looking at other aspects of your developer workflow. So if you're using your own continuous integration workflows, maybe with something like GitHub Actions, in addition to using the Bioconductor build system, GitHub Actions makes it really, really easy to run things on multiple different combinations of operating system, maybe different dependency versions, and to run things on every push. And that's probably overkill, right? We can probably get good results without needing to run things on quite so many different combinations or quite as often. If you use AI coding assistants, it's also possible to configure these in ways that are more efficient, so ensuring that simpler models are used for simpler tasks, and making sure that the agents ask before doing things that are more computationally intensive. Carbon awareness, to be honest, this comes up less often. Most of us don't have much control over the source of electricity that's used for our computing. But if, for example, you're using cloud computing, you could choose a region that has greener electricity. Or if you're using HPC, you could choose to run your jobs at a time when the electricity grid for that HPC is greener. So, you know, if you've got lots of solar power, then run things in the middle of the day. And if you've got not a lot of solar power, then run things in the middle of the night when there's less pressure, less demand on the electricity grid in general. But I think the bigger impacts can come from looking at the user-facing package code. And the principles that are relevant here are hardware efficiency and energy efficiency. So hardware efficiency, this might look like optimizing the memory usage of your package to reduce the memory usage. But it might also just mean documenting your package really well and ensuring that users know how much resources they need to request on their local HPC or on cloud computing to make sure that they're using your package and they're using those hardware resources effectively, that they can, for example, make effective use of any parallelization strategies that you've implemented. Energy efficiency mostly comes down to code optimization, which is a bit more complicated. So I'm going to spend a little bit longer talking about this. So in order to optimize your code, you first need to know where the opportunities for optimization are through profiling your code. So there's a nice package called profviz that can help with this. This is a really simple example of using profviz to profile creating a long integer by concatenating new members to a vector. This is obviously a deliberately inefficient example to demo the profiling. And if you run this in RStudio, it pops up an interactive widget, which looks something like this. So you can see here, does this laser work? Yes. You can see here that almost all of the time is taken in adding new elements to this vector. The initial vector creation is so fast it doesn't even show up here. But you can also see that it's profiling memory allocation and release of that memory allocation, and that some of this time is spent in the garbage collector. And that's because every time you see to add a new element to this vector, it's creating a new copy of X. Okay, so yes, this is obviously not an efficient way to do this, but hopefully it demonstrates what the code profiling output looks like and how it can help you spot areas for improvement. Once you've identified areas where you might be able to optimize your code, actually doing that optimization is going to depend a lot on what your code looks like, what it's doing, but a few places to think about getting started. Vectorization and avoiding making copies of data are always good things to do. And again, many people, many packages are probably already looking at these things. You also, if you've identified a particular function which is slow, then you might look at finding a faster existing function. So a classic example would be fread from the data table package is much faster for reading large files than read CSV. You also might look for a function which is more specialized, that does less. So if you've got a matrix and you're passing it to a function that first coerces it into a data frame and then back into a matrix at the end, maybe you should just look for a function that works on matrices and avoid having to do these extra steps. There's obviously some caveats to optimization. So it can make your code harder to understand sometimes. It can increase the maintenance burden if you're adding additional dependencies or custom implementations. So it's important to avoid premature optimization and to use your time wisely. Spend your time focusing on the areas with the biggest impact. I don't want you to leave thinking you have to optimize everything or it's not worth optimizing anything at all. It's absolutely fine to just focus on the things that will have the biggest impact for your package. So that's been a very quick introduction to why I think this is important and some ways you can try to make your code more sustainable. So if you're feeling inspired, after this conference, you could start by reviewing your development workflow, documenting how to use your package efficiently so your users can make best use of it. You could profile your package code, or you could try doing one optimization. And the online version of these slides has some links to resources that can help with this. But I also have, as part of this NetDrive funding, I have some funding to spend some time helping to work on some of these package improvements. So if you're interested in collaborating or just discussing more, please get in touch. My email is on the slides, or just find me over the next couple of days of the conference. Thank you.
Thank you very much, Liz. Questions on sustainable computing? I'm checking also if we have online. There is.
Thank you for the talk. I was wondering if there is any tool or places you can benchmark, like you can hand in your package and it gives you a rough idea of, like sort of like this library that you showed, but maybe like a more integrated, like you, yeah, give the workflow and it gives you back like a score or something that you can gauge. Yeah.
That's a great question. That sounds like a great service. I am not aware of anything that does this already. What I would suggest would be running your basic work, your typical workflow, maybe from your vignette through profviz, through profiling. That would be a good place to start, I think. But yeah, I don't think there's a sort of a service that does this in a way that gives you those kind of polished results and pointers. Not yet, anyway.
Hi, as someone who's done a whole bunch of high throughput computing, one of the problems that you end up facing is actually data transfer. So if you're passing things around all over the world, like it gets costly. So are there systems for managing what that cost is, should you be trying to pass your compute to somewhere where your electricity is cheaper?
That is also a great question. This is definitely a discussion I've had at some of the sort of environmental computing sustainability meetings I've been at. At the moment, I don't think there's an easy way to measure the relative contributions of, passing data around versus what's saved. I think you have to think about how big is your data versus how long is it going to take to run whatever analysis you're doing. Every situation is going to be a little bit different.
Thank you. And the final question, while the next one is preparing, we can already set up the slides. Do you think bioconductors should consider sustainability for software contributions?
I think that would be great. For me, I've come to see thinking about sustainability as part of a set of related best practices alongside reproducibility, publishing things openly, and so on. And I think there's actually a lot of overlap between the things that contribute towards making software better in general and things that contribute towards making software more environmentally sustainable.
Okay, thank you very much. Important topic. And I'm sure we can continue a lot of these discussions on the coffee break and later. Our next speaker is Laurah Nyasita Ondari, bioinformatician and founder of Bioinformatics Hub in Kenya, which is a collaborative non-profit organization promoting bioinformatics in Kenya. And we will now hear a talk about promoting bioinformatics training and bioinformatics research in Africa. Welcome.
Thank you so much for that introduction. Yes, my name is Nyasita Ondari, and I am a bioinformatician, but also if you're on the Bioconductor Zulip or on the support site or have subscribed to the Bioconductor mailing list, you may have seen a message from me, and that's because I work closely with Maria on social media communications and community engagement for Bioconductor. And today I'm going to be giving a talk on expanding bioconductor training and community in Africa. So what we've learned from our training activities in 2025 and the plans we have for 2026 through to 2027. Okay, so about 20% of the world's population resides in Africa, and Africa has the youngest population in the world. And with that, there is incredible talent, there is rich cultural diversity, and also a growing number of researchers who are developing solutions, not just for African challenges, but for global ones as well. But despite this growing potential, there is still a gap because about only 10% of scientific research outputs comes from Africa. And that is partly because of a gap in capacity. So over the last decade, as Helena said, there's been increase in high throughput sequencing, and that has increasingly become accessible in Africa. So researchers are generating a lot more data than ever before. However, even though there is increase in data generation, there's still a skills gap in analyzing and generating innovative discoveries from this data. And part of that, of course, is because they lack skills in analyzing that data. So despite there being an availability of powerful open source tools like Bioconductor and Galaxy, there are still people who are not acquainted with this and do not know how to use that. And that is where the Bioconductor Africa initiative that we've been working on comes to contribute. Sorry. So to address this challenge, Bioconductor applied and was awarded the Chan Zuckerberg Initiative Essential Open Software funding. And this was to address three main challenges in capacity. So the first one was to coordinate in-person training across East and West Africa. The second one was to increase sustainability of instructors so that we have people who can teach these skills to other people through carpentry certification and membership, and also to partner with key institutions that are regional leaders in training and research efforts, and we have representation of that in different countries, including representation in the African Bioinformatics Institute. So how did this look like in practice? In 2025, Bioconductor delivered 3 workshops in Africa, and a couple of instructors who are in those workshops are here in the room with us today. So the first one was delivered in Nairobi, Kenya, and that was in March of last year. And then another one in Addis Ababa, Ethiopia in August. And the last one we had was in Abomi Kalavi, Benin. This workshop followed a similar format. So we started with introduction to R and the bioconductor ecosystem because we were attracting applicants from a vast background who had different expertise levels. So it was important for us to introduce them to the bioconductor ecosystem to understand how it works and sort of how they can leverage that in their research. We then proceeded to give workshops in RNA-seq data analysis. And then the last day we had a bring your own data. This was particularly important for participants to be able to apply directly the skills they had learned to their research and to their work, and also for instructors to be able to see what are the challenges that people face as they're analyzing data in their institutions and communities. So overall, we trained 83 participants across the 3 workshops, and we were very pleased to see that people self-reported increased proficiency in R and confidence in analyzing their data. A majority of the participants said that they were very satisfied with how the workshops were in and how they were delivered, rating them as very good or excellent. And all the participants that we trained say that they would recommend this training to their institutions and communities, showing that this was a worthy cause for Bioconductor to run in Africa and to see how people were just uptaking those skills. Training participants is important, but also to ensure that that goes on over decades, even after this funding comes to an end, it's important to also train instructors. And Bioconductor has silver membership with the carpentries. So thus far, 6 African researchers have been able to take these carpentry certifications and are certified carpentries instructors. Right now, 3 more are undergoing this training, and we hope that we can keep having more people undergo this training to be able to teach more people, because the scaling strategy that we are thinking about here is that you train instructors, those instructors are able to go back to their communities and research institutes and give the same training to other people. We then create this regional network of instructors who can then go ahead and keep teaching, and we create this multiplier effect. So from the trainings, we also got repeated themes that people wanted, because in as much as we were doing like foundational skills and RNA-seq, people have complex biological data that they also want to analyze. So these 5 key themes were repeatedly asked for: single-cell transcriptomics, population genomics, metagenomics, multiomics, integration, and computational workflow management. And to respond to this demand, this is what we are looking at delivering in 2026. So having advanced workshops, this looks like doing advanced seminar series on these on-demand topics online so that we can be able to reach more people than even what we reached through the workshop. Because we only did it in 3 countries, we have 55 countries in Africa, so we still need to get to the people in the other 52 countries. This also looks like Bioconductor having key partnerships with institutions that also offer these advanced trainings. So thus far, we introduced the Bioconductor Africa Seminar Series. This was launched in April, and we've had people from this Bioconductor community present in those workshops, including Tuomas, who gave a series in May, and we also had Kevin present there as well, although he's not here with us. And although the Bioconductor Africa seminar series is tailored for people in Africa, by Africa, we've been very pleased to see that people around the world are registering for these sessions. And this is particularly important because it connects people in Africa to the wider bioconductor community, which will foster collaborations and knowledge exchange. Yeah. Yeah, and also something that has been a bottleneck to training is not just availability, but also costs. And thankfully, Bioconductor has a partnership with Physalia, which is able to offer two free places rather, to people from low and middle income countries. So far, through this partnership, 108 applications have been received, 8 places have been awarded, and out of those 8 places, 5 people have been from Africa. These numbers may seem small, but that multiplier effect again, when people are able to access these advanced trainings, they can be able to train other people in their research institutions and communities. So what we've learned is that communities are important. Partnerships are also really good for allowing that training and sustainability in Africa. So we are very open to collaborate in case anyone has a package that they'd like to demo with our community. Please write to us. My email is right there, or you can also write to Maria, who's the community manager. Thank you.
Thank you so much. Do we have a question? I may have one. Do you think there are some special aspects in the training in Africa that you are taking into account compared to other regions?
Yes, that's a good question. We tend to see that in-person trainings work much better than online ones. Of course, not just because of infrastructural gaps, but people respond much better because they're able to be in person, to work out their exercises and their learnings and apply them immediately. Yeah.
Thank you. Other questions? I have seen a big rise in the activities in this continent recently, so it's a very bright future for Bioconductor there. We will now have thanking for the speaker and all the speakers. And we will now have a coffee break. Thank you very much for the first session, and be back by 11 o'clock. Thank you.
Let me start. Good morning also from my side. I would be chairing the next session. We have 7 speakers here. It's again in the format of short talk. So that means 9 minutes plus 2 minutes of questions. I would sit somewhere here, and for the last 2 minutes of your speech, I will stand up. We have the first speaker was Elena Zuin. She's coming from Italy, from University of Padova, and she will be talking about batch correction methods and about the new package, which is called BatchF, if I'm not mistaken. So, please.
Okay. Good morning, everyone. I'm Elena. One of the aim of single-cell RNA sequencing analysis is the cell type identification. More cells we have, more we can understand the biological composition of a specific tissue. However, multi-samples dataset can introduce a batch effect. There are technical effects. If we don't remove them, the risk is to misidentify the cell types. In the literature, there are many batch effect correction methods, and there are already benchmarks. So why we need another benchmark? The existing benchmarks have limitations. The first one is about the quantitative metrics used to evaluate the performance of the method. These metrics are inconsistent. That means that for the same method, we have different evaluations. Then the authors use different groups of metrics, and these metrics are included to compute the overall one, even when are not applicable for a specific method. The second limitation is the guidelines. The guidelines proposed by the authors are different because the performance of the batch effect correction method depends on the data characteristics. This is my desperation when I discovered this. So our goals are to solve the quantitative metrics limitations, highlight the strength and the weakness of the method, and try to build a guideline. In this slide, I reported the workflow of the benchmark. We have multiple steps, but don't worry, now I will explain you one by one. First, we select 130 real-world datasets with different biases, such as different organisms, different sequencing technologies, different time points, and so on. Then we extract data characteristics, for example, number of cells, number of genes, number of batches, and we select the most representative ones. Then on this data, we computed the principal component analysis to evaluate how data features contribute to the observed variation. This is the result. So we visualized the first two principal components. Each dot represents the data set. The arrows represent the loadings of the PCA and represent the most representative of features dataset. And with this plot, we can see the association between the variables. For example, the number of batches is orthogonal to the sequencing depth. On the right, I reported the same plot, but colored by the organism, the technology, and the protocol. And you can see datasets with the same technologies are close to each other. Then we consider different batch effect correction methods. We have method in R, method in Python, based on different mathematical approaches. We computed them for each dataset. A method works well when removes the batch effect while preserving biological signal. To solve the limitations, we consider the most used quantitative metrics to evaluate the performance of the method, and we select those that capture independent aspects of the data. So we compute a principal component analysis on the metrics for each dataset and each method. That's why we have a lot of dots. The arrows represent the quantitative metrics that we consider. And you can see we have the IASW, that is integrated average silhouette within, and the Wasserstein distance, which quantify the batch effect removal. These two metrics are orthogonal to normalized mutual information and the adjusted range index, which measure the preservation of biological signal. But we decide to ignore the silhouette because this metric doesn't work very well for single-cell RNA sequencing data. So we decide to use the Wasserstein distance to quantify the batch effect removal and the adjusted range index to measure the biological preservation. Then we compute the geometric mean between these two metrics, and we consider the method with the highest value of geometric mean. On this data, we computed a classifier based on a support vector machine algorithm to delineate the regions of strength and weakness of the method within the two-dimensional space. This is the result. So each dot represents a real world, and the color represents the best method for that dataset, of course, obtained by the support vector machine. And you can see we don't have one method for all datasets, but we have multiple ones. And also you can see there is some white area that represents that for that dataset, the batch effect correction is not necessary. We decide to add some simulations, and this is the result. But last question. For each dataset, is there only one best method? Spoiler, no. So we decide to compute the distance between the geometric mean of the first two best methods. In this case, a low distance value indicates that the best two methods have similar performance. And you can see we have only few areas with a very bright blue color. Last, I want to present you an R package called BetChef, available on Bioconductor. It can be used as a guideline to identify the optimal method. In particular, this function computed the features of the data on your data and predict the optimal method using the support vector machine algorithm, and also visualize the prediction of the two-dimensional space with the red dot. Also, with this package, it's possible to use the method I consider for the benchmark and also compute the quantitative metrics to evaluate the performance. I want to thank you for your attention.
Thank you, very interesting. Can I ask, I don't know if I understand correctly, so batch correction usually happen before cell annotation in single
That's
cell.
right.
So
yeah.
what kind of biological variation you are evaluating in your benchmark?
Okay. When I consider public datasets, in this dataset there are the labels. So I trust the labels inside the data. I consider the labels to evaluate the biological signal. I know that it's a little bit, yeah, but yeah.
So with batch correction, we always talk about how we want to get rid of the batch effect, but sometimes we overcorrect. And I see that's not something that people look at. So in your package, do you have any metric to measure if you're accidentally overcorrecting and getting rid of signal?
Well, consider the combination of a Wasserstein distance and the adjuster index. I consider the overcorrection situation.
So in your data set, you mentioned that you had human, mouse, and multi-species.
Multi-species mean human and mouse.
Okay, so it's not
Yeah.
actually multi-species; it's human
Yeah.
and yeah.
Yeah.
I was wondering, like, if you account for different, let's say, for example, I work with plants, and then if you try this method in plants, they fail miserably, all of them.
Yeah.
So yeah, and then yeah, so the benchmarks are always based on human and mouse.
Yes.
I was wondering, like, if you had tried this.
Well, for now I consider only the human and mouse, but I have a question for you. There is some good data set for plants?
A lot.
A lot,
dataset,
okay.
Yeah.
Okay.
And the other thing is, if you're doing a similar question to what the colleague asked her, if you're doing this for, let's say, the same tissue across different labs, if that is the batch effect you're trying to correct for, or if you have atlas level batch correction as well. So different tissues from different labs.
Well, I consider different tissue, different disease. I try to consider different biological situation.
Thank you.
Thank you. Really nice and interesting talk. So you had a slide where you kind of color-coded the distance between the best and the second best method.
Yeah.
Lots of data sets, doesn't really matter whether you take the best and the second, and some data sets were very blue, which means that there was a big difference between the first and the second.
That's right.
Do these data sets have specific characteristics that you identified or you showed in the very beginning? So could we anticipate, based on the characteristics of a data set, that this is one where it will maybe not really matter, but this data set has characteristics where I will have to be really careful which one to use?
It's a good question. I don't check the characteristics of that this data set, but I cannot answer, but I will check. Thank you.
We would have also 1 minute for maybe one question from the Slido. So maybe the first one on the top. Is it possible to use for different data types, like maybe metabolites? Oh, it was now shift down. Sorry. Sorry.
The first one?
I mean,
no.
Sorry, it was shifted down. It can be used to a different data types. Maybe, for example, metabolites. It's moving all the time. So because your method is used for the single cell.
Yeah, it's for only
single. So the question was, there was also another one, if it can be used maybe to bulk RNA-seq or maybe metabolomics, or is that somehow which is fixed only to?
For now, I only single cell RNA sequencing data.
Okay, so thanks a lot. Thanks again. So the next speaker is António Sousa. I think he's a local person here from Turku, right? And he will also talk about integration of different data sets and about a package which is called Coralysis. Sorry, it doesn't work. Can you screen?
Good, thank you. Yes, so my name is António, and I'm a PhD student here at Turku Bioscience Center in Professor Laurah Gatto's lab. And first of all, I want to thank the organizers for giving me the opportunity to present here the main work of my PhD, which consists in the development of our bioconductor package called Coralysis for the sensitive integration of single-cell data. And this work has been published last year in Nucleic Acids Research, and it's since then available on Bioconductor. Integration, as you heard previously, consists in identifying shared cell types across heterogeneously generated single-cell data sets. So giving a set of data sets that could have been generated, for instance, in different laboratories or using different library preparation methods, here representing different colors. Ideally, you want to jointly analyze these together in order to identify the same cell types, and you want to avoid the situation that you see represented here, where you end up by having multiple clusters per cell type, and you want to integrate this data in order to get, let's say, 3 clusters, 1 cluster per cell type. Our solution was to develop this multi-level integration algorithm, which we implemented in Coralysis, which basically progressively integrates cellular identities through multiple rounds of divisive clustering, as you can see here. At its core relies on a logistic regression model with L1 regularization. And basically, as you saw, across the different clustering rounds, it finds the smaller or fine-grained populations in the data and improving the mixing of the cells coming from the different batches. So this is a single-cell RNA-seq data set that has been sequenced in 10x 3' using different assays. And this is just a t-SNP projection highlighting here the dataset identity and the cell type composition on the right side. So Coralysis performs integration. That was the main aim that was developed for. And as you heard in the first talk, there is also this reference mapping where you can train a data set and annotate that data set and then use that reference to basically classify new data sets and transfer the labels from that reference to new data sets, avoiding the kind of cumbersome of annotating the data set novo. So it allows us this reference mapping. And I will not have time to go into this, but the software gives basically a set of clusterings and a probability score of a cell belonging to each one of these clusters. And this probability score can be used at some extent to infer cell states. Because, for instance, if you have a cluster and a set of cells that are assigned to that cluster, the cells with the highest probability are probably the best representatives of that cluster, meaning that they do express, or they tend to show that they do express a set of markers that are very good and representative of that cell or of that cluster cell type. While the cells with lower scores, usually what they tend to represent is somehow a cell that is maybe transitioning. For instance, if you have B memory cells and B naive, it might be some cell that it's somehow memory, but maybe does not express so well these markers. So at some extent, you can use for that purpose. We benchmarked our method against top performer integration methods in one of the benchmarks that Elena in the previous talk mentioned from Fabian Terry's lab. And we saw that our method performed comparably well with these methods, and we saw that it could offer a benefit or outperform others when integrating cell types in imbalance context. So imbalance is basically when we have a cell type that is more abundant in one data set than the other, or in this more extreme case that I'm represented here in this UMAP is where we selected 2 cell type pairs. So this is PVMC, so essentially immune cells from blood. We have a control sample resting PVMCs and STIM, which are basically activated or immune activated with interferon in vitro. And here it's the batch, and here it's the cell type. And we selected 2 pairs of cell types. So in this case, there is a CD14 monocytes and CD16 monocytes. And basically, we kept one in one of the data sets and exclude it from the other, and vice versa. And we did the same with CD4 naive and memory T cells. And what we wanted to see is if the methods would over-integrate these similar cell type pairs or not. And Coralysis, as you can see, was able to keep these apart and at some extent integrate relatively well most of the other populations, maybe not a bit not so well here, for instance, as you can see for these dendritic cells, but overall it did perform better than the other methods, and it kept the CD4 and naive and memory cells apart, and CD16 and CD14 apart, while for instance other popular methods like Harmony on single cell BI kind of mixed these populations, as you can see there. Coralysis was originally developed for single-cell RNA sequencing data, but it also is able to integrate other types of data, single-cell proteomics. Here I'm bringing an example again with whole blood, but site-off data, so 39 surface markers or protein markers, not sure if all of them are surface. Basically, the two data sets are samples from patients with influenza, and the others have been stimulated in vitro again with interferon gamma. And as you can see, this is how the data looks before integrating, and after applying Coralysis, you can see that Coralysis was able to identify and align the two data sets quite well, even these basophils that are quite low abundant in the data set that are represented there. And this is not just based on visuals, but as you heard in the previous talk, so this is based also on quantitative metrics. And overall, you can see that this batch correction, aggregating these batch correction metrics, you heard like about some of these before, as well bioconservation. So there is a slightly improvement, at least, so it doesn't mess with that, but there is like a considerable improvement of batch correction. And as I mentioned, it also performs this kind of reference mapping. So to highlight this, I brought here an example where there were a set of data sets of PBMC, so these immune cells from blood, that have been sequenced in different or using different library preparation methods. We select one of these as a reference. The reference here comes from a 10x genomics preparation method. And what we did was then we mapped or we predicted classifications for the rest. And ideally what we want to see is that this is a perfect mirror of this. Obviously, as you can see, it's not, but it's still fairly good. So this is the predictions, this is the ground truth. And overall, the accuracy was decent. And you can see that there is a decrease based on the technology as expected because the reference was at 10x. Then we tried to understand why it was so well in the seq-well. And basically, the method was able to predict two cell types that were originally missing. So one is this tiny population here that has been originally notated as CD14 and actually CD16, as you can see by the absence of this marker, but the presence of this gene alias for the CD16. And the other population that we are able to identify is these NKs that are here close to these cytotoxic CD40 cells, as they are transcriptomically similar. And again, they miss the expression of CD3, which is a marker of these CD8 cytotoxic T cells, but they do express this marker of NKs. So I think my time is up. So I can conclude. I acknowledge and thank my lab and also the funders. Thank you.
Thank you very much. Very nice talk. I'm wondering how Coralysis deals with cell types that are absent in one of the batches, or even several cell types, you know, that may be present in one batch but not in the other. Like, are they mixed in with the most similar ones, or are they kept separate? Or
in the example that I show, it was able to keep them separate. But obviously, I mean, I guess it depends on case by case. But so the reason why it worked so well is because so it relies on this logistic regression with L1 regularization. So internally, the software and algorithm does feature selection and tries to identify the communities itself. So and that's one of the reasons why it tends to perform better than other methods. That, for instance, because some of these methods, what they do is they do the integration in the low-dimensional space. So they try to align the cells on the low-dimensional space, and obviously they try to find these neighbors in this space, and they look to the most similar cells across the data sets. So they, in a way, they are not looking anymore to the gene expression feature, while Coralysis, it's always looking into that.
So it's a really nice attempt or a good approach to think that doing it based on cell type. But you should use the immune cells, which have pretty clearly defined populations. How does this work in other diseases, like, for example, cancer, where you can have all sorts of different mutations, all sorts of things going on, especially across different patients who may have different subtypes of that cancer? How does this work in that environment?
I don't have that much experience running on cancer. Yeah, so I cannot really comment on that. Sorry.
I'm sorry, for the sake of time, we have to move on to the next speaker. Thanks a lot again. Okay. So the next speaker is Beatriz Campillo. Who, despite this being a European conference, she's coming from US, so welcome to Europe. And she will be talking about how to interpret the splicing data, if I'm not mistaken. So if everything works.
Thank you. Okay. While we set up, I didn't make it to the hackathon, but I know some of you guys were there, so you may know me as the person who printed the Tidiomy stickers. So if you want to come by later and find me and get some of them, I'm happy. And also we have some other from the lab and also for the splicelogic package, so you should definitely come get one of these. Okay. Okay, so I will go ahead then and start. So I'm going to be talking about splicelogic, which is a new package from the lab to annotate the splicing events from isoform structures after doing differential transcript usage analysis. So I come from University of North Carolina, Chapel Hill, where we work with a lot of RNA labs that study splicing, which is basically the mechanism that splices out introns to form mature mRNA molecules. And this is regulated in part for with RNA binding proteins that I will be showing in some examples later. Where is the laser here? Later in some of my use cases, but basically through alternative splicing, a single gene may produce many different isoforms, and the changes in the proportions of these isoforms between different conditions leads to what we call differential transcript usage, and these may include changes in where the transcript starts or ends, or in the sequence between exon-exon junctions, and is also influenced by cell type, environment, drugs, genetic variants, etc. So to perform differential transcript usage analysis, we part from RNA-seq data. So this new package, splicelogic, where does it fit into the RNA-seq workflow? So after we perform transcript quantification, that's when we can go ahead and perform DTU analysis with, we can do this with many different bioconductor packages. And then here, when we want to perform post-DTU analysis is where splicelogic falls and to perform exon level events. Alternative splicing has different types of events. And in SpliceLogic, we have specific functions to detect each of these, for example, cassette exons or skip exons, and so on. And we also have a wrapper function if in case we wanted to call all of them at the same time. So now I'm just gonna go ahead and explain what SpliceLogic basic uses each. So we can imagine a situation where we have an experimental design in which, in the presence of RNA binding protein, a specific exon gets spliced out, so it's not present in the final isoform. But if we knock down this protein, this exon gets spliced in, and we see it in the final isoform. So if we perform RNA-seq with this, we will have our set of transcripts, and then we can go ahead and perform the DTU analysis again with any set of methods. And from either of these, we would obtain something like a coefficient that basically would let us know if the isoforms changed in proportion significantly, and whether this went up in the contrast condition or if this transcript went down in the specific condition. So this is what we essentially need for SpliceLogic. We would need essentially, we can do this manually or with a prepare exons auxiliary function that we have to basically obtain a G-ranges object with the exons of these transcripts that change it each proportion and their DTU coefficient. So then we would call what we've called the preparesys function, that is just simply used to harmonize the input. And after that, we can call one of the find functions that I've introduced at the beginning. For example, if in this case we call find all events, in the example that we are thinking about, we will identify this skipped exon event, or this we would identify these two exons that are mutually exclusive. So the output of any of these find event functions is another G-ranges object with the exons that are involved in a alternative splicing event, and these new annotated columns let us know the type of event that the exon is involved in, and also it's important to keep track of this new event TXID, which is the transcript relative to which this event is happening to, because we can imagine in the situation or the simply example that we have that we only have two transcripts, one that went up and one that went down. It's kind of trivial to know which one is, but in the case that we have more, it's important to keep track of it. So that's essentially the output of SpliceLogic. So we have developed SpliceLogic to be fully vectorized so that we can easily work with all transcripts of all genes at the same time. Is supposed to be readable, modular, and since it uses Bioconductor objects at input and output, we hope that is easily integratable into already existing workflows. So now I'm just going to present some use cases that we can maybe use SpliceLogic for. So I know I've been talking about DTU the whole talk, but what if you don't want to do DTU? Just what if you just want to compare two sets of transcripts first? So we can imagine a group where we have a set of transcript in our reference, and then a set of transcript that we've detected to be novel, and we want to see how are they different and what alternative splicing events they have. So we can simply use another of our auxiliary functions to prepare the input and generate that G-ranges object that we've seen at the talk. And then maybe we want to find all the events, and then we can easily pipe this down and pull the event type and maybe visualize a little summary of what we find. And maybe we see that, oh, like half of the events in my new transcripts are detected to be skipped, for example. We can also maybe, if we go back to our RBP example, wanna analyze what is the sequence that this RBP is binding to. So, for example, we can, after finding, if we wanted to focus on skipped exon events, we can maybe first visualize the results that we have and verify that essentially we are actually obtaining events where these exons are getting skipped. And then since, again, we have a G-ranges object, we can pipe this down and maybe flank upstream of these exons and get their sequence and visualize the signals that we have. And we have done this with GTEx data from the protein PTBP1. And as you can see here, we were able to retrieve that the signal is there since this RBP is known to bind regions rich in Cs and Us, and we were able to see this in the upstream region of the skipped exon events. And then finally, maybe instead of going about the RBP, maybe we want to see the proteins impact about the events that we see. So again, we want to focus maybe on the skipped exons, and then we would easily retrieve the TX ID, like the IDs of these two transcripts, and then maybe analyze the domain impact of the skipped exon event, or maybe we can take this sequence and then go ahead and perform 3D structure prediction, maybe with AlphaFold, or and that's it. I want to thank my lab and hope that you guys try it out, and maybe if you have ideas, inputs, or issues, reach out to us through our GitHub. Thank you.
Any questions from audience? Let me start. So do I understand correctly that you have to always overlap it with some annotation? So what are the possible sources of the annotation? I mean, I saw that you are using the TXTP, but what are the other possibilities? Can you also use the, whatever, I don't know, ensemble and whatever?
Yeah. So as long as you are able to retrieve the sequence, and so the input is a G-ranges object, so no matter like what method what method you use to create the G-ranges, that's totally fine.
Okay, perfect. Yeah. We have some questions on the Slido, if there is no question in the room. There is one.
Integrate this in any NF4 pipeline. There is one pipeline in NF4 for this differential exon splicing, alternative splicing.
So I don't have any plan for that yet, but this is a very new package, and that's kind of our hope to start maybe using this, trying it out, and integrate it into already existing workflow. So if you have ideas, or we can talk more about that, because this is the whole point. Thank you.
There is maybe one more on the Slido. I mean, did you benchmark your tool against other existing tools?
I have not benchmark this yet against other existing tools. I would love to hear if you have inputs about which other tools, because we, as far as we know, this is one that
I guess it's quite unique, right? What you have for, I mean, all the other tools are steps before.
There is a isoform switch analyzer that would be the closest to us, but we have not found any that we would be able to actually compare it to. But I would love to hear about them and then benchmark it.
Cool. Thanks a lot. Thanks again.
Thank you.
You're on the next one, right? So we have our next speaker. I guess I don't have to introduce him, but maybe at this point I would like to say maybe we should clap one more time for Tuomas, because he was one of them, really like the leading person organizing all the things here. So thanks a lot, Tuomas, to bring us here.
Hop. Yes. Thank you for kind words. Yes, you might wonder why I'm wearing this. So the reason is that we are all superheroes because we are doing open science. Yes. All right. So about the presentation. Well, oh yeah, I have wrong. So I was changing slides in my computer and didn't realize that it's have to do like this. All right, now I'm back in the track. So as we know, in Bioconductor, the data containers are in core. Everything starts with the data containers. And if we agree with this certain data format, data structure, then developers can develop these interoperable methods utilizing this data container. And what that means ultimately is that then there are lots of different methods to use for the user when they put this their data into this specific data container. So everything starts with this data containers, right? Again. So the summarized experiment is the main data container in Bioconductor. It was developed for this generic data object for biological data analysis, and now it is used in metabolomics, proteomics, microbiomics, genomics, and you name it. It's used in all these fields, and that is why it's the one of those most downloaded packages in Bioconductor. But but microbiome field, they were using a different data object called a phylogic, for instance. It's very good package, and they have awesome community there, but the main problem there is that they are not using the same data structure as others. It's like dancing in a corner while others are dancing in the dance floor, the disco dance. If you are in Tulip, you probably see that video of disco dance instructions. And because of this exact reason, Huang et al. Extended this summarized experiment with these three structures. And we need these three structures, for instance, to phylogeny trees, which is the usual format to represent these bacterial hierarchies. And this is now the core in microbiome research in Bioconductor, and that's the most optimal data format. But this data structure alone, it's like a car without the engine alone. It's like a Ferrari in a garage, but you cannot drive it. So that's why we build the methods, the engine. We build the basic common operations that people do usually in microbiome research. And for instance, this MIA package is relatively used nowadays. And then one big thing is also this online book, which has been trend in Bioconductor. So first comes the methods, then comes the online book, so that everyone can see how the methods are actually used and share the best practices in the field. And there is now this preprint available. But of course the work is not done. Yes. So we have this common data standard, tree summarized experiment. Then we have the interoperable methods, which are not only developed by us, our team, but also by the international collaborators. So there are lots of people that are now utilizing this tree summarized experiment structure. Then we have the shared knowledge, the online book, and we have a way to collaborate with each other. But we have these, but I truly believe that the next challenge is not technical. The next challenge is to build the community around this everything. And of course we have the ways, the tulip and so forth, but still people are not collaborating enough in microbiome field. And what I truly see up is the Tidiomics people. They have been very successful building this very nice community where they are sharing information and collaborating with each other. And that is something that I want to see also in microbiome field in the future. And this is the reason why I created last night this pull request of microbiome working group in Bioconductor. Nothing is official, it's not accepted, but I just created this pull request. So this is just an announcement that there is something has been done. And the idea is now that we create this microbiome working group in Bioconductor, we have a couple of project ideas, end-to-end workflow for microbiome data science, and then carpentries training material, for instance, developing them. And everyone is welcome to join this working group when it is accepted. So that is the main point, that now the idea is to create the Bioconductor, this working group. And I want to finalize this. I want to read this. It's bolded here. So the software is important, but the community is essential. That was great words. Thank you for your time.
Thank you. So any questions from the audience? I see already one hand.
I really like your introduction on data structures and methods. Therefore, I would like to hear what's your take on the absolute mess that there is on the, how to call it, like object-oriented approximation in R, where you have, the S3 as based, S4 mainly in Bioconductor, S7 coming, R7 done independently.
Yeah, well, in Bioconductor, it's we have this. Well, there is this goal of interoperability. So we have this summarized experiment, for instance, but the idea is not to be alone in the corner, but create methods to interoperate between, for instance, Python and other languages, and also other ecosystems in R. So I lost the question, but that it can be a mess, but Bioconductor tries to make it as smooth as possible. Yes.
Any other question?
Thank you. So great talk. Great idea to have this community. I was just wondering if you, let's say, focus, let's say, quote, only on Bioconductor and R, or are you also trying to get, I don't know if there is Python community on microbiome or other software out there. Are you also trying to get those to work closer together, or any ideas?
Oh, well, good idea. And I welcome you to join the working group.
Thank you.
So if there are no more questions, I would suggest we just move on because we are a little bit behind schedule. Thanks again. So the next speaker is Alex Cecchetto. Yeah. Perfect. He is also coming from University of Padova, from Italy, and he would be talking about clustering gene expression based on the spatial latent traits.
I use this thing. Yeah. Hi, everyone. I would like to start by thanking Francesco Denti and Davide Risso, who have this joint work with them. And spatial omics technology allows us to measure molecular on the interstructure of the tissue. And during this work, we will use this data set that is a colorectal cancer biopsy with 4,000 spots and more than 18,000 genes measured, obtaining a large matrix to study how tumoral stroma, epithelial, and immune population are structured in the tissue. And biologically meaningful gene features vary across the tissue, and that's why it's really important to handle this dependence. But to do that, we need a model that is flexible enough to handle this spatial dependence, but also scalable enough to work with high-dimensional data. And that's why we introduce our model, where we model counts with Poisson likelihood, and the expected gene, the expected expression is linked to a linear predictor where we have different source of variation. We have a baseline with an library size offset and a gene-specific intercept. Then we have gene-specific covariates. And finally, we also have a low-rank matrix factorization term, and that here is just used to model the residual variation that is not handled, not modeled through the fixed effects. This is nothing new because it's something that in single cell analysis we are already seeing, like GLM-PCA, SGD-GMF, or ZIMB wave. But none of this actually take into account the spatial dependence of the spatial, how to handle the spatial dependencies. And that's why we thought about performing a spatial functional regression by using basis function as covariates in the model, and have given a set of basis function phi1, phi k, the underlying spatial effect can be modeled, can be approximated through the combination of this basis function, linear combination of this basis function. Here we have an example of B-splines function at this different resolution, and as you can see, coarser bases can catch a more broad spatial trend, while more finer bases can catch a more specific variation into the tissue. And we work in the Bayesian paradigm because we're working with high dimensional data and spatially structured. And the Bayesian framework allows us to regulate, to act regulation method to, because we're working with a lot of high parameters. And moreover, we have, through the posterior computation, we have an estimate of the uncertainty of the estimates. And we place weakly informative Gaussian priors on the model parameters. And we do that centering them on 0 because they act as a regulation, but we also allow them to have large effect if they are supported by the data. And as I was mentioning before, we have the last part is the latent, the low rank latent term is used to capture residual variation. And we don't want it to be confounded with the basis part. And that's why we projected the latent factor U onto the complement, the orthogonal complement of the basis space. And finally, we also use a lower triangular structure on the loadings factor V to avoid the rotational ambiguity and to enhance interpretability and the stability of the factors. The interesting part is that we performed also a Bayesian clustering on the gene-specific effects. We use a stick-breaking prior there is a flexible way to infer the size and the weight of the clusters. working with the discrete cluster membership can be hard while working with the gradient-based algorithm. That's why we use a Gumbel softmax relaxation that basically is a differentiable approximation to obtain the cluster membership. And finally, we place also priors on the mean and the scale parameter for each group. We place a Laplace on the mean to enhance a shrinkage on the parameters, and an exponential on the scale factors to allow them to be positive and also regularized. And that's how we obtain a cluster-specific distributional profile for each group. After the model is defined, the inference is performed in NumPyro. There is a probabilistic programming framework built on JAX, and it enables automatic differentiation, just-in-time compilation, and GPU-accelerated inference. And we do that using variational inference with a mean field Gaussian approximation to obtain a tractable approximation of the posterior distribution. And for each latent parameters, they are represented by a independent Gaussian distribution. But basically, the output of this is posterior summaries for the main components of the model, like the latent factor, spatial coefficient, mixture weight, and cluster membership. And here we plotted the posterior spatial prototypes that basically they summarize the spatial signal that each component is able to, let's say, catch. Each one of them represents a mode in the spatial variation in the tissue. And through the stick breaking prior, only 4 groups were actually active. And here we summarize the biological signal by plotting the mean log expression of the genes associated with higher probability to each cluster. And we can compare the signal with the observed annotation of the tissue, like how they are able to find a meaningful signal. And then we also compare the computational time of working on the CPU and GPU in different scenarios. And we can see how the GPU implementation, working on a GPU becomes always more necessary while the dimension of the data increase. And as future direction, we want to use a multiple resolution spline to be able to find, to model both a broad and a specific spatial variation. Then we want to use the residual latent matrix decomposition, for example, to inform spatial annotation. And finally, we are working on the developing of a methodology into a bioconductor package to enable reproducible inference, visualization, and interpretation of this data. And I'm happy to take any questions, suggestion, or sorry. Thank you.
Thank you. Is there any question from the audience here? James here.
You talk about a method to impose I think a lower triangle. There's some sort of method used for improving identifiability using a lower triangular representation on the matrix B.
It's something that in the Bayesian literature they do. Like when you do matrix factorization, you just impose the loadings to have a lower triangular structure with a positive diagonal.
Oh, okay.
It's something. Yeah.
Any other question? I guess this is going along the same thing. The question here. What was the package in Bioconductor? You said you are only...
we are working on that, yeah.
It's in Python implementation at the moment.
Yes, yes.
Okay. Any other question? No? And again, for the sake of time, I will suggest we move on. Thanks a lot.
Thank you.
So the next speaker is Luka. University of Glasgow, right? And would talk about the rhinovirus annotation, right?
Thank you, everyone. So as introduced, I'm Martha, coming from University of Glasgow, and I'll talk to you about, I'm shifting slightly to virus genomics. So I'll talk to you about a new package which enables reproducible genotype assignment. So a brief introduction to the virus. Rhinovirus is very common within the human population, and actually anytime you have the common cold, it's most likely rhinovirus. But more severely, it's been associated with asthma and chronic obstructive pulmonary disease, and also more severe lower respiratory tract infections. And genetically, it's a positive sense single-stranded RNA virus belonging to Picornaviridae. And we currently have over 169 genotypes, which are actually antigenically different. And this explains why children can get up to 12 episodes of the common cold within a year, because infection from one serotype does not protect you from the subsequent one. And this becomes very important because when you begin to study the dynamics within the population, it's like we need to understand how many genotypes are circulating before we can make any transmission inference. So here I show you the genomic organization. It's about 7.2 kilobases, so it's very short. But of particular interest, I want to draw your attention to the VP42 region and the VP1. So either of these sections can be used to assign the virus to genotypes. And the two regions have been shown to show congruence. The VP42 is slightly shorter, so it's most commonly used and was our basis for forming the package. So this is overall a slide showing you how we previously used to genotype the virus in my former lab. So we start with generating the sequences, the partial sequences, and then we can, once we call the consensus, we align them using a tool of choice, and then we use another tool to calculate pairwise distances, maybe mega or something else. And then after that, we now end up with this distance matrix whereby we have a pairwise sequence of each to each. So it's quite broad. And then we already have a currently established genotyping approach to it, whereby these researchers collected all publicly available sequences, and then they did a pairwise calculation of each to each, and they drew this histogram, and the 10.5, for example, is the lowest dip, as you can see from the zoomed in figure. So that's the lowest point, and we assume that separates the intra-type from the intertype distances. And so with that, then you can create custom scripts to then be able to assign your newly generated sequences to specific genotypes. But one challenge you observed was that when different members of the lab tried to do this whole process, a few sequences would become mismatched. So one person would assign a sequence to S62, another would assign to A23, and it was not clear where this disparity was coming from. And so that is why we came up with this package, rhinotypeR, and it just brings the entire process under one roof. And so we have two options whereby you can start with downloading the prototype sequences, which are embedded within the package, and then use your alignment of choice. For example, if it's outside R, and you can align your sequences alongside the prototype sequences, and then re-import them into R. Alternatively, you can just import your newly generated sequences into R, and within R, we can combine them with the prototype sequences and then align them. And so with that, the main function of interest is actually assign types, which now is able to use to perform a pairwise distance calculation of each to each, and then after that, we can be able to see. You can do multiple things, such as see the frequency of the types, but also we have other functions that can support your epidemiological analysis of this virus. So such as pairwise distances, and then calculate, be able to visualize how this varies. And this is just how that would look like in real life. So you can start with your newly generated data, and then once you have assigned your sequences, you end up with a table that looks like that. And you see that even sequences that do not meet a very strict threshold, you will have it as unassigned, but you will have the nearest reference. And so maybe you can take that sequence and do further analysis such as look at recombination. Maybe it's very diverse from the original prototypes and things like that. And you can also visualize the frequency. And then in addition, you end up with these histograms that you can look at the identity of sequences and a simple phylogenetic tree. We also have this sneak peek function, which you can identify a sequence of interest and look at mutations of subsequent sequences relative to that. So it's just good to see where the mutations are happening within the virus. And so we now wanted to see how our package compared to other tools, and we ended up generating this huge distance matrix across these three tools of interest, and we were able to get a really good correlation of almost 1. So that's to show that at least it's working well. It's in terms of distance calculation, we are getting it right. And in terms of time, it was relatively fast, so 1.41 seconds, which is slightly slower than ape, but takes about 1/10 of the time that Mega would do, and this is for a typical data set. And the additional time it takes is because we have extra checks within the package just to make sure that everything is in order. So future directions, as I mentioned, we currently have 169 genotypes. These are continuously increasing, so it's always important to keep track of that. And also now, this is based on the VP42 region. There is people who prefer to use the VP1 because it's longer and more informative. And also there's a interest to have this in Python as well. And so thank you very much. This is the associated publication.
Any questions here, Fabrizio?
Thank you for your talk. So very central to how you classify to how you genotype your data is the measure you use to measure distances.
Yes.
The pairwise distance measure. How do you do this? Is it model based? Because usually you try a bunch of models and then you use model selection software to find the best model, and based on this model, get distances.
Yes, so we have different, at the moment you have the ability to choose a model that you want to calculate your distances on. You can choose to go, for example, because there are different things that inform your model selection. Maybe it's data collected within a very short time, so you don't expect to be a lot of evolution within the sequences. For example, you can go with a simpler model, but data collected over a longer longitudinal study would need a different model. So we allow that. The simplest model, we also have the simplest model, which doesn't take into account any evolution. So you just do p distances, which you can start with and maybe compare to more complex models. But we have all that ingrained. We do not go about to guide one on how to choose their model because we feel like it's quite complicated.
Yeah, that's why I was wondering why you compared with Ape and Mega. These are all tools that don't do model selection. So
no,
yeah.
but you can pick, for example, you can pick models in Ape and Mega as well. So for example, you can do with for the figure I just showed, we used the simplest model across all. So we picked the same model across all.
Yeah, but again, users have to know. They have to specifically say what model to use.
Yes.
It's not like chosen based on data,
No. But we picked, because it was the same data set and the same model across all, we just wanted to see, are we calculating distances the same way as existing packages?
Thank you very much.
Yeah.
Thank you. There are no more questions here, then I would suggest to move on. Thanks again. And our last speaker of the session would be Sophie Marie Wind. She is from University of Minster. And she will be talking about comparison of different tools doing a 4C analysis and coming up with some consensus, right?
Yeah. So let's see if I get this right. Yes. Thank you for the kind introduction, and I'm sorry for being the last person between you and your lunch. So today's talk will be about 4Synergy, which is my PhD project. And I will give you first a short introduction into the world of chromatin. So we are interested into the spatial organization of chromatin, of the genome, because deregulations in it can lead to deregulations and therefore to alterations into dysregulation and diseases such as cancer. But the good thing is that alterations are potentially reversible. And for CSeq is a sequencing technique which allows us to identify exactly those chromatin interactions. So our biological aim is biological aim is to find chromatin interactions, while our technical aim is just to find peaks in the coverage track. Genau. And yeah. Oh. Yeah, there are several tools available to fulfill exactly this task. We have Forseca, Pixie, R3CSeq, and Forseq. And a benchmarking of all of those tools revealed that none of the tools performs adequately for all main use cases that we have in CSeq analysis. And therefore, we decided to develop a new algorithm. But we decided to go for an ensemble algorithm because we have enough individual tools and they are diverse enough for this task. So we decided to go for a weighted voting-based approach. So what we are basically doing is we have our training data, then we are running all the individual tools. We get results vectors out of the tools, and then we are combining those results vectors with corresponding weights. And using then different performance, predictive performance evaluation metrics to optimize the weights on it, and our result is then to optimize weighting. But this will not be the focus of today's talk. Next step was, okay, we need to see if our approach works. Therefore we did an benchmarking of our tool, our ensemble tool called ForSynergy, across the existing CSeq tools, and we can see that in both F1 score and AUPRC, we were able to outperform the tools. So we did this on only 20 data sets, but we needed data sets with a ground truth, and they were super hard to find. So it took us 1.5 years of literature research to get this set of data sets. Now we will come. So now the question is, how can we bring our ensemble algorithm to the user? Because it's not that trivial as, for example, just bring one algorithm to your user. We have to bring 5 algorithms to the user. And therefore, we built a ForSynergy framework. So first part of the framework, which can be split into 2 parts, is a pre-processing pipeline. Because we always have a standardized pre-processing where we need to homogenize all in and outputs of the tools. Then we have to run all CSeq tools, so Pixie, R3CSeq, Forseq, and Forseca. And all of those tools are written in different languages. They have different dependencies and so on. And then we have a second part of the analysis, which is the custom analysis with the ensemble algorithm. And we want to have a user-friendly and customized analysis with different analysis modes. That's why we decided to go for Bioconductor. So we have the framework consisting of a snackbag pipeline because we need the reproducible and scalable pre-processing. And then we have the customizable Bioconductor analysis. And we have a Shiny app running on top of it. And now I will show you the interplay between snackbag and Bioconductor. So if the user wants to run the pipeline, the user first needs to download the pipeline. Then he needs to provide the input data in a config file. Then he needs to run the snackbag command to start the pipeline. The pipeline is then writing out all results that we need, so quality control and CSeq-based analysis. And then we are coming to the Bioconductor package. So we have a constructor called createIA, which is then creating an object of the ForSynergy class and loading all data that are created by the pipeline into the object. And then we can run our ensemble algorithm using consensus IA. Then the user can decide if he wants to use an F1 optimized or AUPRC optimized model from the optimization I showed you earlier. And then we also offer a differential analysis mode where DSeq is run in the background. Yeah, and of course we have visualization functions, but I did not show them here before because for the technique they are not that important. This brings us to the last point. The challenges if we want to integrate a snake-make workflow, I mean, you could also integrate a Nextflow workflow, this doesn't matter, into a bioconductor ecosystem. So I would say all of those points are not big challenges, but you should be aware while implementing it. So first is the architectural compatibility, because, for example, in SnakeMake, we have this directory structure, and this directory structure should not collide with the bioconductor directory structure. We have description namespace, the R directory, and so on. So they should really not collide, as long as you're using one repository for both. Then this was not a big point for us, was the dependency and environment management. Since we decided to go for an conda environment per rule, it was all stable, and we tried to keep the bioconductor dependencies as minimal as possible. So this helped a lot, and therefore this was not a big problem for us. Yeah, then another point is the build stability and the runtime constraints. I feel like this was a little of a bigger point. So usually packages should build in less than 10 minutes, which is quite limited, and therefore you should always go for smart examples. Yeah, maybe it's also connected to the point of sustainability into package development, because we should be smart and use really small examples that we can stay under this 10 minutes. And then the question is, okay, how we want to bring it to the user. Do we want to have one repository for the pipeline and bioconductor, or do we want to have two repositories? And we went for two repositories. So the advantages of going with one repository is that it's in one place and easy to access for the user. But on the other hand, we have no independent versioning and no independent releases. Means if we have an update in our pipeline, we need to make a version bump, which is maybe not always what you want. The good thing about two repositories is that we have a clear separation, so the user gets not distracted, and we have an independent versioning and releases, which is quite good. I would say disadvantage would be on the first side, okay, we have two repositories, but I would say as long as you document everything well, this is not a problem. And yeah, I'm at the end. Thank you. That's it from my side.
Thank you. Any questions from the audience?
Hi, thanks for a nice presentation. I just have a question about the last bit about the two repository. I have seen a lot that in this type of kind of paradigm, people usually neglect to report everything or version of everything. So I wonder if there is any kind of automated way in your pipeline so that it already saves all the version information so that the user, don't need to be worried or distracted about it.
So no, we just did it in two repositories, and therefore we thought that this might be a clear enough separation, and we have, I would say, quite comprehensive documentation that this should be, in our opinion, sufficient, if this answers your question. Sorry, yeah. Yeah, we thought about adding a function, and we will maybe add a function in the future because it would be smart to just run the pipeline by having a function run workflow. So we will add this, but I decided against it because of the checks and so on. And because we also have a Shiny app, which is not part of the bioconductor package. And there, one part of the Shiny app is the kind of creator for the snake-make command. So this is part of a Shiny app, basically.
Thank you for the presentation. I don't really know snake-make, but I was wondering why you decided for it instead of like targets, the like natural R workflow orchestration package.
So that's a good question. In the past, I usually worked with Nextflow or snake-make for workflow management systems because it's more polyglot, I think, and therefore I was from the first moment on, I was like, okay, I will do it in SnakeMake, and yeah, just so the decision for me was SnakeMake on Xflow.
So if there are no more questions, I would suggest we thanks the speakers from the session again. And before we leave the room, I guess there's some additional information.
Yes. Right. I am not a chef, but I truly believe that part of this conference thing is to experience the culture. So I decided the foods. So today is very Finnish day. This is very like a typical Finnish food. And then dinner will be the archipelago style. Very good. And then tomorrow, the Sweden is well known for the meatballs, but we have meatballs also in Finland, and I think they are better. And on Friday, for our Italian friends, we have our version of macaroni things. So macaroni laatikko. All right. Then yeah, the lunch is next to the info desk. Then about the workshops. So please take this QR code or then go to this web address. So these are the divisions. So Guillem is then the president. In ministry there will be Ilaria, and Tellervo there will be Stevie. So I will let you to copy that web address. So those rooms are very close by. If I wear my Crocs, I will be in the 2 seconds from one